Okay, this is second try at making this lecture, this time with audio, which I've double checked and should be working. This is our lecture on genetic engineering and bacterial transformation, leading up to our bacterial transformation lab that we're going to do at the end of this kind of unit, whatever you want to call it. And so this lecture is going to do a couple of things. It is going to kind of walk us through some of the most basic background information on DNA. Since this is our first lab, we kind of got to start at the beginning, and then a little bit of the historical background of transformation, like what does it mean, and finally a little bit on the lab itself and what we're going to do. So since this is not me actually giving the lecture, it should actually go a little bit faster because I don't have questions to deal with in class. So okay, starting off, we have got to kind of go back to the beginning with double helix, right? And so if we were to draw DNA like this, if you're ever going to draw a DNA molecule, you've got to draw the double helix like this. So it's got these portions on the outside and then portions on the inside. So think of a twisted ladder. It's always how we describe it. And I'm going to kind of go quick through some of this, mostly because everyone in this class has already had ninth grade biology, and some of y'all have had AP biology. And so I'm not trying to go through everything all at once, right? Okay, so there's a couple different ways that you can describe this molecule, right? This molecule can be described as being double-stranded, right? Double-stranded implies that, well, there's two strands. That one's really easy, but the strands themselves, each one is made up of, it's a polymer of what are called nucleotides. A nucleotide has a very specific structure, right? And so this is a nucleotide. There's an oxygen right here, and we get 3 parts to any nucleotide. This is a deoxyribose sugar. Now, if you don't remember, like any angle has a carbon here, we don't draw the hydrogens. And so this is a 5-carbon deoxyribose sugar right here with an oxygen holding it together. Think of glucose, but with 5 carbons instead of 6 and a different structure, but it is a sugar, a deoxyribose sugar. At the top right here, we have what's called a phosphate group. There's an oxygen, a phosphorus, an oxygen, and an oxygen, and an oxygen. Because of all those oxygens, it's super negative charged. And up here, we get one of 4 different nitrogen bases, right? So we can put a little flag around that. Now, I'm not going to ask y'all about the structure of those, but that right there would represent one of these, what we kind of, if you want to simplify, little L-shaped things right there, right? But the other side of the DNA molecule, on this side, right, it is its own polymer of nucleotides, right? So we're going to get to the other part of that in a second. So you can call it double-stranded, and it's a polymer of nucleotides. You can also call it anti-parallel. Anti-parallel, and in the simple version, we can put just a little 5' here and a 3' here. Follow the 3, and we get to a 5' here and a 3' here, and we can see that they point in opposite directions. But what those terms actually mean has to do with this sugar molecule right here. Since it's kind of the center of the molecule, I'm going to put an H right there. We can use it to reference direction, right? So on a clock, if this right here at the oxygen is midnight, then this is the first carbon, this is the second carbon, this is the third carbon, this is the fourth carbon, and then this angle is the 5th carbon that the oxygen is attached to. So what that means is on this side over here, that's the side with the phosphate pointing up, right? And on this side over here, on the complementary other anti-parallel strand, that's the side with the oxygen and the hydrogen off of the sugar pointing up, at least in our picture right here, right? So they point in opposite directions. So double-stranded, they're polymers of nucleotides. Anti-parallel is 5' to 3' and 3' to 5'. And then, of course, we have complementary is our last way of describing the DNA molecule, right? So the DNA molecule is complementary, which means we're talking about the bases, right? The bases here can be adenine, thymine, guanine, and cytosine, right? And so those bases will actually form hydrogen bonds, right? And that's kind of important, in between other bases, but only if they fit and match up together with positives and negatives and electronegativity. And so let's say that this right here is, I can kind of write it on top of it. Let's say that this is a G right here. So that's a guanine, right? Well, complementary to that, and the only one that will fit is a cytosine, right? So there's a cytosine. Now, if we make this anti-parallel, this goes up, so this goes down, so this has to go up, and then we go this way, and this is always kind of hard for me to draw like this. So there, and then the phosphate is going to go this way, and oxygen, phosphorus, oxygen, oxygen, oxygen. There we go. We got it. We have a base pair. But only the cytosine will fit, and I could show you pictures, but it's not really what we need to worry about. Like, why does it only fit? But when it does fit, it forms 3 hydrogen bonds right there, right? So 3 hydrogen bonds between guanine and a cytosine, but between adenine and thymine, we only form 2. There's a major difference there, right? 3 hydrogen bonds versus 2, and this can go back to one of the first experiments that studied and learned about the chemical nature of DNA, right? Because depending on how much of a DNA molecule is GC, you can figure out its GC percent. That affects its melting temperature, or the temperature at which these hydrogen bonds right here will break, right? And so this guy, Edwin Chargaff, he figured out using the melting temperatures of DNA that maybe in humans, adenine could be, say, and I believe these are the real numbers. I think I looked them up last year. Adenine could be 28.5% of DNA, right? Well, then he could figure out thymine, guanine, and cytosine. And so let's say that this is human, right? Well, if we know that adenine is that, then we know that thymine is as well, because for every adenine, there's a thymine. And so we get that. And then I believe if I do this right, let's see, 28.5 and 20.5 is 56, 57, leaving us with 43, which would be 21.5, right? So we get 21.5% here, 21.5% here, right? And so he figured out the GC ratios, or what we call the base pair rules, adenine and thymine, guanine and cytosine. But he did this with lots of different organisms, right? So he could have done this with like a mouse, and I'm pretty sure these are not the right number. I'm just going to make these up, right? If adenine is 30%, then I can automatically know that guanine is 20%, because this has to be 30%, and that means that's going to be 60. Leaving us with 40 divided by 2 is 20%, right? So today, in a lab setting, we might use GC percents in order to measure the melting temperature of DNA. And there's all these little formulas to do this. The formulas change depending on the length of the DNA strand. So that's really kind of easy to do. It's literally just like algebra 1. You have a variable, you solve for it. But one formula is this. GC, or the melting temperature in, by the way, degrees Celsius, is equal to 70 plus 0.47 times the GC percent, right? So I can't remember I think this is for larger strands. I have one that's for like 10 to 14 base pairs. I think this is a little longer than 14 base pairs. But we could take the GC ratio and we could plug in, let's see, we could plug in the GC ratio of 41 or 43, I guess that is. 43 times 0.47 plus 70, and that's going to give us the temperature at which this DNA strand will separate, because you've put enough energy into the molecule to break all of the hydrogen bonds in that. So that is how you calculate melting temperatures of DNA. We're going to do that a little bit more in the next lab. It's just an example right now. That's in the PCR lab. We'll come back to that. All right. By the way, here is a picture of that, right? If you take, this is kind of on its side, but I like this one because it shows the rings here. And so you can see that like adenine is a 2 nitrogen ring structure, and thymine is a 2, and you get 2 base pairs. Guanine is a 2 ring, and cytosine is a 1. So you get, and then you get 3 hydrogen bonds. This idea that there's actually 1, 2, 3 nitrogen rings across, 1, 2, 3 nitrogen rings across, 1, 2, 3, 1, 2, 3. It's one of the ways that the cell knows how to, like, when it's replicating DNA or whatever it happens to be doing with DNA, it knows which base goes, right? It has to fit the width of DNA, and it also has to fit the right number of hydrogen bonds at the right spot. Okay. Next up. How do we know this, right? We got to ask the question of, well, we do know the structure of DNA, but how? And that gets into photo 51. This is photo 51, and we talked about this a little bit in the historical perspective at the beginning of the class. And so this is a photo taken by Rosalind Franklin, and she was an X-ray crystallographer, so she was really good at creating crystals of organic molecules, and taking X-ray, doing a technique called X-ray diffraction of those molecules, right? And so we take this laser, this kind of X-rays really, and it's got a string of DNA that is held up in this sample right here, and then it would be in a vacuum chamber. You can't have any air in there. You shine the X-rays at it. Some of those X-rays are going to pass straight through, and some of them are going to hit atoms and bounce off, right? And so my analogy of this is like, imagine a very dark room, and there's a chair in the room. You don't know what it is, but it's a chair, and you want to know what's in the room with you. So you start throwing tennis balls at it, right? And the only way that you can figure out what it is by throwing tennis balls at the chair and then seeing where they bounce. You know, it would be very, very difficult, but if you did it enough, you might be able to figure out, based on where the chair was hit by a tennis ball and where the tennis ball landed, what the shape of that thing is. And so this is the photo that she took, and then James Watson and Francis Crick took this photo, and they did some interesting math with it, which I do not understand, but apparently this is explaining some of it, right? Like the distance between the top beam of electrons and the center is the distance between base pairs, and the missing layer line equals more than one strand wound around each other asymmetrically. So I can't do that, but they apparently knew that the helix diameter is 20. Was that 20 angstroms, I guess? And so that tells you, like, I don't know how you can figure that out right now. I don't know, but it's possible people can do that by doing math and figuring out like how to work backwards, right? So that's what they did. The combination of all of them told us that that's what the DNA strand was. Okay, next up is gene expression. And I know that in most of my classes at this point in time, we will have already done an activity. So I'm going to kind of go pretty quick here just to try to remind you guys about some of this stuff. So if we take this piece of DNA, right, and this piece of DNA like this, I'm not going to be very careful with my double helix, right? So let's say this is an entire chromosome, one of the human 23 pairs of chromosomes. If it's chromosome 1, it's about 200 or so million base pairs. And so then we can highlight like a region of it and say that this is a gene. It's actually more difficult than you think to define gene because of the way that gene structure works, right? Like, where do you define the beginning and the end? And it doesn't really make a lot of sense. And I'll explain why in a little bit, but let's say that this is a gene. This gene is a store of information. In my freshman class, we call it blueprints, right? Like, it's the blueprints for building a house, but it's not the house itself. It's the information to build it. And so this gene is going to be transcribed. There's an enzyme called RNA polymerase, and so it kind of sits on here on one of the strands, and it unzips DNA, moves in that direction. And as it does that, it's going to read it, and it's going to build this molecule. This is messenger or mRNA. There's a couple of stages here that I'm not going to really talk about. They're called mRNA processing, introns and exons, and 5' caps and poly A tails. And they're really interesting. AP biology specifically covers a significant amount of that information, but we don't in here. That's all right, right? So this is now an mRNA molecule. Now, we can look at it right away and see, oh, it's single-stranded. By the way, it has a uracil and not a thymine in it, right? And so it's a chemical difference. Uracil is not as chemically stable as thymine is. And so DNA has to be a chemically stable molecule. So uracil wouldn't work. It would break down too fast. But this is not the final product, right? We have to actually take this molecule, and I'm going to have to redraw it over here like this. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. So I picked 18 of those bases right there. And there's organelle. The organelle is going to fit on here. And let's see, 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6, 7, 8, 9. It's going to fit on here. And there's actually two parts to this organelle. Like this. It's a ribosome, right? So this is a ribosome. So a ribosome is an important organelle. It is not a membrane-bound organelle. It is a soluble organelle that is a combination of catalytic folded ribosomal RNA and protein. Really complicated biological machines here. And they make protein. I'm going to come back to this here in a second, but prokaryote and eukaryote, they have different ribosomes, and those ribosomes are defined by the two parts to them. Think of it like a hamburger. That's a simplified explanation, but these two parts are named after their density in a sucrose gradient. It's this kind of like high-tech way of measuring the density of small molecules. Regardless, it doesn't matter, but there is a 30S, that stands for a Sveberg unit, and a 50S, and that's the small subunit and the large subunit, and they combine to make the 70S ribosomal, which is the whole thing. Those are not additive because of the way that sucrose gradient density centrifugation works. Like, again, we're getting into like ways of measuring density of very small molecules. It doesn't matter, but they don't add up. So eukaryotes, though, have different ribosomes. 40S plus 60S equals 80S. Those, let's come back to that in a little bit, okay? All right, so the whole point of this is that the ribosome reads the mRNA, and as it does that, it will fit onto the mRNA these other molecules known as tRNA right there. And so the tRNA right here has an attached amino acid on it. And so once it has read that one, another one is going to fit in. So we'll put the other one like right here, right? And then the actual chemical change that happens here as the ribosome moves and ATP is hydrolyzed and it is used to change the shape of the ribosome and all that kind of stuff that happens, it will actually take this and attach it, forming a peptide bond right here. And then as the ribosome moves, this tRNA is no longer needed and another one can show up. So here's the next one. They have names for these. It's called the access site, the peptide bond site, and the exit site right there in the ribosome. So it's like a three-step thing: access, peptide, exit, and then slide one more. But the final product of what we call translation is this, is a chain of amino acids that folds up into some sort of functional protein that does something for the cell, right? And so that's the final product. That is an expressed gene. Now we can go back and we can come up with some sequence here. Like, let's just make a sequence. TATGGA, TACGAC, CTAGGA, ACCATTCCA. This is a sequence of DNA, right? I know that because it has thymine in it. It will then be transcribed into some sequence of an mRNA, right? Whatever that happens to be. And then it will be translated, but only from where it is at. Actually, let's say this whole thing right here is transcribed, right? The entire thing is the mRNA molecule. But it'll only translate from this AUG to the UAA, the stop codon, right? So that's the coding sequence of the gene right there, using the genetic code that I know we're all familiar with, right? All right, DNA, RNA, protein, gene expression, and I want to transform bacteria to express glow-in-the-dark genes, and so we need to take the DNA that makes them glow in the dark, and we need to get that into the bacteria, and then they will produce a glow in the dark protein that they will then glow, right? Okay, I said I was going to come back to this. This is kind of important, right? Because we are about to use antibiotics. The fact that we have different ribosomes means we have a target for treatment for bacterial infections, right? Anything that is unique and different between bacteria and our cells means we can try to stop that thing in them and without hurting us. So it turns out this right here is a target for antibiotics. We use antibiotics a lot in this class. A target for antibiotics, right? Prokaryotic ribosomes, because if you can inhibit it with some molecule, you can stop them from making protein and they're going to die, right? Now, there's lots of different types of antibiotics. This specifically is tetracycline, and its derivatives is it inhibits their protein synthesis. It doesn't harm us at all. Now, it doesn't distinguish between beneficial and pathogenic bacteria, but our ribosomes are perfectly fine to continue making protein in the presence of tetracycline, no problem at all. We're not going to use that. We're going to use ampicillin, which is a penicillin derivative. I'm pretty sure ampicillin is a cell wall affecting antibiotic, but I'll confirm that when we're doing the lab. Okay, next up. So I'm not going to spend a ton of time on this because we have already done the DNA to disease lab, but like the reason it's hard to, so we could take this, we could transcribe it and translate it, right? I'm not going to bother with that. But let's see, this is the whole gene right here, right? The reason it's hard to define a gene is because like where does it begin and where does it end? So let's say that this region right here is the transcribed region, right? So that is what is going to be the mRNA. And then within that, smaller than that, you have the region between the start codon and the stop codon, and that is the coding sequence, right? So you have in the mRNA, you have some upstream 5' untranslated RNA, and you have some 3' downstream. We use those terms a lot, upstream and downstream untranslated RNAs after the stop codon. So we would translate that region in the coding sequence if we wanted to know, and it would be from the start codon AUG to UAA, the stop codon. But what about this stuff, right? This is right in front. These are known as promoters. These are important in recruiting to the area RNA polymerase. So there's certain proteins that bind to this. It's called the TATA box. It is a conserved sequence that is a promoter. There's the GC box that's 85 base pairs upstream. There's the CAT box. There's all these sequences that can show up in most genes and some of them in all, something has to be there in all genes that are going to trigger expression and regulate that gene expression of that specific gene, especially in multicellular organisms where you have differential gene expression in different tissues. So those promoters, are they part of the gene? Like, yeah, they are, but they're not part of the coding sequence or the transcribed sequence. So yes, they are part of the gene. There's even other sequences that are like super far upstream from that called enhancers that can be as much as like, I don't know, 10 kbp, 50 kilobases, like that's a long way, 50,000 base pairs upstream from the gene. And it's still interacting with these promoters as a gene sequence with transcription factors. And again, we're getting into differential gene expression, and I don't necessarily want to, but just to know that promoters are important. So if we're going to transform bacteria to glow in the dark, then we need to include a promoter on that, and we will. So we'll talk about that promoter. All right, we need to compare the genomes really quick because if we want to understand transformation, we got to be able to compare these. So that's the human genome right here, right? The human genome is 3.2 billion base pairs in length. That is, by the way, 3.2 gigabase pairs in length, right? Because that's just me. That's billion right there. You can have megabases, kilobases, and gigabases, and it's just an easy way of kind of saying things instead of like dealing with lots of zeros. We have 23 chromosomes. That's 22 autosomes and one pair of sex chromosomes. And within those 23 chromosomes, we have 20,000 genes, much less than we thought, especially when you start comparing it over here to the bacteria. In the late 90s, when the Human Genome Project was occurring, and everybody thought there were going to be hundreds of thousands of genes because we're just so complex. And well, it just turns out there wasn't, right? So all of this is what we consider the human genome, right? But we actually have to multiply this times two, because, well, we have two parents, right? So mom gives us one whole set, dad gives us one whole set, and so we've got a lot of DNA inside our cells. I've read that it's like 7 feet of DNA inside every single cell. I don't know if that's one copy or two. Now, of this, all of this stuff, only 1 to 2% is coding regions. Regions where the DNA is actually coding for a protein. So a ton of it is transcribed but not translated. A ton of it is promoters. A ton of it is enhancers. A ton of it is pseudogenes or viral like remnants or telomere, highly repetitive telomere sequences, or highly repetitive centromere sequences that don't code but are important in cell division. It used to be called junk DNA. We really try to avoid using that term because it does have functions and roles. It's just not that it's going to make a protein, not going to be coding for an amino acid. All right, let's compare that to our bacteria here. By the way, all of that is in all of these little chromosomes that you can see during mitosis. Right? Okay, here the DNA is a lot different. There's one chromosome and only one chromosome. One circular chromosome. So the DNA connects back to itself, right? It makes a ring of a double helix where it connects back to itself. If this is, say, E. coli, it is, and I looked this up, 4,639,675 base pairs in length. We could average that up to 5 megabases, right? So that's a lot smaller than ours, right? But they're single-celled prokaryotic simpler cells. They don't have multiple chromosomes. They have roughly 4,200 genes, and that's crazy. People thought that was gonna. I mean, that's like, a little less than a quarter, maybe a fifth of. Yeah, it's a little more than a fifth, a little less than a quarter of the number of genes in a human. Right, that's crazy. But it just turns out there's ways of increasing complexity that don't involve more genes. And take AP biology if you want to learn more about alternate splice variants and all that kind of stuff. Also, just regulating gene expression at a more complicated level and having different tissue types can add in complexity. So now the important part for us is that there's one other thing. There is plasmid DNA. Plasmid DNA is this. It is small. It is circular. It is extra chromosomal, and it is non-essential. So that means a couple of things. That means that there's a lot you can do with it. Like, we can use it in the lab. It's so small it doesn't fracture and tear when you're trying to pipette. There's a ton we can do with plasmid DNA, and that's what we're going to do. All right. Zooming forward to this historical perspective, to understand transformation, we got to go back to 1928 before we knew what DNA was, and we got to go back to Frederick Griffith. He liked injecting mice with bacteria, right? He was actually not a DNA scientist because that didn't really exist at the time. People didn't know about the double helix or the role of DNA, other than it was like cells were full of it. And he was asking the question of, why do some bacteria infect and kill mice, but others don't, like trying to understand why we get sick, right? So he starts injecting mice with bacteria, and the first one he injects is the rough bacteria, and that mouse is perfectly fine, right? No issues at all with that mouse. That is a non-pathogenic bacteria, so it wasn't hurt at all. He takes another strain of that bacteria that looks different under a microscope. It looked smooth, so he calls it the S strain, and that mouse died, right? And so he's trying to ask the question of, what is it about this S bacteria that kills the mouse, whereas this R bacteria doesn't have it? So he takes that same S bacteria and he kills it, right? So this is S bacteria, but it is heat killed He basically boils it for a long time. And when he boils it, all the bacteria die. He injects it into the mouse, and unsurprisingly, the mouse is perfectly fine. It doesn't die, right? The neat thing that he did, the thing that made this a really famous experiment that we still learn about today, is he mixed these bacteria together. He must have let them sit for a little bit. He didn't know what was going to happen, right? Now we follow his procedures basically to do this, but he took live S plus heat killed R bacteria. He mixed them in here individually. The mouse is fine. The mouse is fine. Mix them together, let them sit, and then inject it. And sadly, the mouse dies. But that shouldn't happen, right? Because it's combining this plus this, and individually they don't kill the mouse. So he tries to describe it. He says that when you take the S bacteria, right, and you heat it up, it ruptures, right? It dies. So now we know it goes through this cell lysis process, right? And so it's, does this like that. So the S is dead and it releases everything. And then over here, if you mix it together with this live R bacteria, something from this dead bacteria is going to still be around, and it's going to get taken up by this R bacteria, which will then transform its behavior, its genetic identity, into a functioning essentially like an S. He called it a transforming factor, right? Now we call it plasmid DNA, right? He said that the R was transformed into an S. Now we call it bacterial transformation. We can transform bacteria because we can trick bacteria into doing this, whatever we want. If we have a plasmid DNA, a bit of plasmid DNA, and we have some bacteria of certain types of time to do this, we can mix the two together and we can change the genetics of a bacteria and transform them with new and novel DNA and genes, right? And that's what we're going to do in class. We are going to transform and trick some bacteria into expressing certain things. Specifically, I've got to get through this one first, right? Okay, there's one other step here, right? Because we don't want to just take some random plasmid from a bacteria. We want to use our own plasmid. The one that we're going to use, by the way, is this. Here it is, pGlow. pGlow is about a plasmid that has been genetically engineered, and it has a couple different genes or important areas in it. It has an origin of replication so that the bacteria will copy it. When the bacteria replicate their DNA, they will, I guess this is actually the origin. I'm not sure. Probably that one right there. They're going to copy this DNA so every bacteria gets it. It has a green fluorescent protein. It has an ampicillin resistance gene that we're going to talk about why that's so important. It has an arabinose gene, which is involved in making a strong promoter that forces this thing to be expressed and make lots of it. And we can turn regular old E. coli into this right here. We're going to transform them. But to make a plasmid like this is not natural. This has been engineered and built by genetic engineers with restriction enzymes. Restriction enzymes cut DNA, and they cut DNA at specific sequences, right? So there's this enzyme known as EcoR1. It was kind of the first one discovered, E. coli restriction enzyme 1, and it cuts DNA at a sequence that is palindromic, GAATTC, and then this sequence is actually cut apart from this sequence right there. Okay, the ability to cut DNA seems like it's not all that special, but if you take a plasmid like this, like that, and if you cut it open, then you can actually have in a test tube this open piece of DNA, right? And then separately from that, if you take DNA from some other gene of interest, let's say that this is human DNA, and let's say that this right here is the insulin gene. Well, we can cut that out with the same restriction enzyme, like this, and then we can mix the two together. We add in some DNA ligase that seals broken ends, and we end up getting a plasmid that is going to have mostly a bacterial plasmid, and then stuck inside it is the human insulin gene. That's genetic engineering and creating your own plasmid. We are not going to create our own plasmid. There's a couple of levels of complexity beyond what we're doing, but we are going to purchase a pre-made plasmid called the P-Glow, and it has all of these options. You can see where it has these cut sites right here that can be cut with restriction enzymes. And so it's got all these different things in it that we need in order to see what we want to see when we genetically engineer bacteria with this plasmid. And that, I believe, is it. So hopefully this helps as far as understanding, one, the background information behind this kind of stuff and what we're going to do in class.